Hey guys, today we start topic 1.4, membrane transport. We're going to split this up into a couple of different parts. Today we're going to talk about passive transport. It's a relatively short lecture, and then you have some simulation work to do. Next rotation, we'll talk about active transport. You'll have notes to do for that and probably a simulation or two. And then we're going to do our third practical, which is an osmolarity lab, and that'll probably kick off Friday. So, if you aren't sure where you should be looking for your skills, understandings, and applications, you should be looking in 1.4. Make sure you're taking good notes so that you can use those on the next skills test. Passive transport for today. This is where we're focusing. We're going to talk about simple diffusion, facilitated diffusion, and osmosis. And just as a review, here we have a model of the cell membrane, right? We have those hydrophilic heads, which are composed of a glycerol and a phosphate group. And we have those fatty acid tails, both saturated and unsaturated. Together, those make up phospholipids. And those are the major component of the cell membrane, along with some integrated proteins, right? Some integral proteins. Okay, so the hydrophilic heads of the phospholipids, the ping pong balls, make it difficult for large or charged molecules to pass through. So if you're small and you're not charged, you're nonpolar, you can pass right through. If you are large, you don't fit. It's not going to work. So we're going to talk about passive transport first, particularly how we get small particles, uncharged particles, across that cell membrane. So understanding number one, particles move across membranes by the first method is simple diffusion. So let's talk about some basic stuff. Diffusion is the passive movement of particles from areas of high to low concentration until equilibrium is released. Anyway, so we talked about this freshman year. A concentration gradient is when you have lots of particles grouped together, and then over time they spread out, kind of like the fart or the perfume analogy, whichever you prefer, right? One person farts on one side of the room. Eventually, even if you're on the other side of the room, you're probably going to smell it. And that's because those particles travel over time. Now, diffusion can happen in fluids. Diffusion happens in gases. It doesn't matter what phase you're talking about. So whether you're talking about in water or you're talking about in air, particles move from where they are to where they aren't. Kind of like your room, right? So like your room doesn't ever clean itself, right? You have to clean your room. We start from highly ordered and we move towards disorder. And that's entropy. That's one of the laws of thermodynamics. And it's kind of just the nature of our universe. This is important when we're talking about cellular processes, right? This diffusion is the result of random collisions between particles, and this requires no energy. No energy. That's why it's passive. And when you're talking about the concentration gradient, we have a high concentration here, right? We go from a high concentration to a low concentration. And that's diffusion. That shouldn't be like rocket science to anybody. We talked about that quite a bit freshman year. Even if you weren't in my class, if you were in Everhart's class, I'm sure, this is old stuff, nothing new. So here we can see this is diffusion in action. We have particles that were in a highly concentrated area. They're spreading out. They're mixing. And that's the result of random collisions between particles. Okay, so when we're talking about cells, it's not that easy. We have a permeable membrane here, right? Semi-permeable membrane, meaning some stuff can get through. These small nonpolar molecules, so you can see, even though water is polar, it can still get through. It's small. It's super small. And it can just diffuse right through the cell membrane. You can see it going back and forth. But these green particles, these are larger particles. They could be polar or they could be ionic, meaning they hold a charge. And that phospholipid membrane is not going to let them pass through. You can see that while the water molecules are passing back and forth across this membrane, the green particles are stuck on whatever side they're on. That's because the phospholipid bilayer is selectively permeable. Some molecules can pass, while others can't. And remember, that's caused by the polarity of the hydrophilic heads of the phospholipids, just so we're clear. Examples of things that can travel through simple diffusion: oxygen, carbon dioxide, other small nonpolar molecules, and water. Now, that makes sense and it's important because you want all of these things to be able to pass through the cell membrane without having to expend energy. These are all things the cell needs, right, to either take in, like water and oxygen, or get rid of. And it has to do that pretty regularly. If you had to spend metabolic energy to move those materials in and out of the cell, you would have to consume a lot more energy. You'd have to eat a lot more. You'd have to eat pretty much all the time. So that's not feasible if you're a multicellular organism, right? So it makes sense that these small molecules that are necessary components, either products or reactants for cellular processes, they can move through without the use of energy. Now, stuff that can't pass through on its own that requires energy, things like glucose, large molecules, polar molecules, anything that's water soluble, ions, things like that. Okay. So simple diffusion occurs when particles pass through phospholipids and diffuse across the membrane without the aid of a protein. So there's no channel here, there's no gate, right? There's nothing helping this stuff move through. It's just these particles, and you can see they can pass right through the membrane from that extracellular space. Extra means outside, right? They're passing on through to the intracellular space, or the inside of the cell, until that concentration is equal. Once that concentration is equal, the particles don't stop moving. They just move back and forward at the same rate. So just as many are going to come this way as are going to come this way, and the net is going to be the same. Remember, you can pause this video if I'm going too fast. You can pause it. You can rewatch it, however you need to do it. The next method that we're going to talk about is facilitated diffusion. So, facilitated diffusion is when you're using a protein channel in the cell membrane to allow particles into the cell. Now, whenever we talk about a molecule that's passing into a cell that's not water, we're going to call it the solute, right? So you can always think of the solute as the stuff that's coming in and out. So here we have extracellular fluid. This is the fluid outside of the cell. This is the cytoplasm inside the cell. We have a protein right here, a transmembrane protein, and we have some solutes. Now, when we're talking about facilitated diffusion, this is charged particles or larger molecules diffusing through channel proteins that are embedded in the membrane from a high to a low concentration. There are more of these particles on the outside than there are on the inside. As such, they are going to diffuse from high to low with the help of this channel protein. So we're still not using any energy. It's just this channel protein is a special tunnel or a special gate that allows only this molecule inside. That's facilitated diffusion. Facilitate means to help. This channel protein is helping this solute diffuse through the membrane. Channel proteins correspond to the specific particle that they help diffuse through the membrane. So, remember, in biology, your form is your function, your shape is your job. So these proteins, which remember, proteins are composed of amino acids. You put chains of amino acids together, which form peptides. Those peptides twist and bend to form two-dimensional and three-dimensional conformation, or their three-dimensional shape, right? They kind of turn into like this slinky. Now, if the channel itself is negatively charged, if that protein has a negative charge, it's going to attract positively charged ions to come through. It's also going to repel negative ions. So, a negatively charged protein channel attracts positive ions. Similarly, if your protein channel is positively charged, right, it's going to attract those negative ions and it's going to repel positive ions. So the specific conformation or shape and specific charge of your protein molecules are going to dictate what they allow in and what they allow out. So, negatively charged interior will only allow positive ions to pass through, and a positively charged interior will only allow negative ions to pass through. And that makes sense. The third type of transport that we're going to talk about is osmosis. Now, osmosis is the passive diffusion of water across a semi-permeable membrane. We call osmosis its own special word instead of just diffusion because we're specifically referring to water as the molecule that's moving. Now, water always goes to where it isn't, right? Like you never spill water into your cup, you only spill water out of your cup. So, water is going to go freely either in or out of the cell, depending on where the highest concentration of water is going to go. Freshman year, if you were in my class, I think we did the egg experiment, right? Where we soaked the egg in the vinegar to get rid of the shell. And then we stuck a bunch of eggs in syrup and those shrunk. And we stuck a bunch of eggs in distilled water and those got bigger. And then we stuck some in salt water and those didn't change at all because they were isotonic. So this is that same concept over again. So we're going to be using the words solvent and solute. And just as a refresher, solute concentration determines the movement of water across the membrane. Water moves from hypo to hypertonic areas. Now, when we say hypo and hypertonic, we're talking about the solute, right? So I use the example of Kool-Aid powder and water. So, or Crystal Light when you're making Crystal Light or you're making whatever like powdered flavor drink that you like, right? So the Kool-Aid powder would be the solute and water is always the solvent. So when we say hypertonic and hypotonic, we're talking about the solute. So hypo means low solute, hyper means high solute. Okay? So water is always going to move to where there's more of it. Here on this side of the membrane, you can see there's water molecules and just a couple of green particles. So we would say, compared to the other side of the membrane, this side of the membrane is hypotonic. And that's because there's less solute, which is the green. And if there's less solute, that means there's got to be more H2O. That's really messy here. It's because I'm writing, my surface is like standing up and I'm trying to write with it standing up. So I'm going to lay it flat and we're going to try this again. Okay. This side is hypotonic. That's a bit neater, right? Because we have low solute and high water, right? The solute is green. The water is the little blue particles. That means this side is hypertonic, right? Hyper means high. I can spell high. Let me try that again. High solute, which is the green, and low solvent. There's less water molecules on this side, which means water, remember, water is the thing that's moving. Water always goes to where it isn't. So water is going to go this way. If you're talking about osmosis, the thing that moves is water. Ooh, that's weird. Okay. Okay. Oh, look at that. Hypertonic, higher solute, lower solvent. Hypotonic, lower solute, higher solvent. Okay. So water movement is going to keep occurring until the solute concentrations are equal. Isotonic. Isotonic means equal. So if you have concentrations that are the same, that is isotonic. Here we have two diagrams. We have the top diagram and we have the bottom diagram. So let's look at the top one first, shall we? Here we have a cell which has 300 solute, or the concentration of solute is 300 on the inside, and the concentration of solute on the outside is 150. Now, water is going to move back and forth because we're talking about osmosis, right? Water is going to move back and forth until the concentration is equal. Oh, okay, so I see. So that means the particles are not the solvent. The particles are actually the water particles, right? Okay. So we have more water on the inside, right? More water on the inside than we do on the outside. Water is going to move out of the cell until the concentration of water is equal inside and outside of the cell. So let's see. Inside the cell is hypotonic, because we're talking about the concentration of water. Outside of the cell is going to be hypertonic. And water is going to move out until the concentration of water is going to be equal. So let's take a look at the bottom. Here we have the water concentration inside of the cell is less than outside, right? So water is going to move in until the concentrations are equal. Now, these diagrams are really meant to show you the cell volume, the size of the cell. On the top one, right, we see that the volume changes. The volume gets bigger. Let's do some examples just to drive that home. Okay. On the left, we see a hypotonic solution, right? Hypotonic solution means that there's more water on the outside than on the inside. So let's see. We're going to use the tables at the bottom to help us out. I'm going to make up some concentrations. If the solution is hypotonic, right? So that means the solution has lower solute concentration, right? So our solute concentration is going to be 10. And if our solute concentration is 10, that means our solvent has got to be 90%. And if you add those up, you get 100%, right? That is a hypotonic solution. We have low solute concentration. My cell is probably going to be hypertonic, right? Now, when I say hypertonic, it doesn't mean that it has to be like 90 and 10. It just means that there has to be more solute than the solution. So I could literally make this, if I wanted to, I could make this 20%, right? Which means that this is 80%, because that adds up to 100%. Now remember, water goes to where it isn't, right? So where is there more water? Remember, water is solvent. There's more water in the solution, which means water is going to go from the solution into the cell. If you can draw this table, if you can mimic this table or some version of this table whenever you get a question like this, you can't get this question wrong. You can always tell where the water is going to go. So if you're an animal cell, that water is going to keep coming in until that concentration is equal, right? But remember, you're a cell and your membrane is pretty flexible. And if too much water comes in, you're actually going to burst. And that's what being lysed is. Like a cell will actually pop. This is why you don't get distilled water when you go to the hospital. You get saline, because the concentration of your blood is not zero, right? There's lots of dissolved stuff in your blood. So you need saline, which is isotonic, equal, right? So that your cells don't burst. Now, if you're a plant cell, you're okay, because the cell wall will prevent the cell from bursting, right? And when a cell is full with water and it's nice and stiff, we call that turgid. So you know how plants get like really wilty when they dry and they don't have enough water? That's this plant over here. But when you give it water, that water fills back up the cells and the plant stands back up again. And we call those cells turgid. Okay, let's do an isotonic solution. So when we say isotonic, we don't mean that it has to be 50% solute, 50% solution. That's not what we're talking about. We mean, how is the outside? It's got to be balanced to the inside. That's isotonic. So if my solution is, let's say, 30% solute and 70% water, right? Isotonic, if the cell is isotonic to the solution, it's the same. 30% solute to 70% solvent. Now these both add up to 100%, so we're not missing anything, right? And the water is the same in both. So water is going to move freely back and forth across the membranes, and you really don't have any change in shape. Plant cells tend to be kind of squishy when that happens. And now